Good morning, ladies and gentlemen, and welcome to the 4BASEBIO investor presentation. Throughout this presentation, investors will be in listen-only mode. Questions are encouraged, can be submitted at any time using the Q&A tab situated on the right-hand corner of your screen. Please just simply type in your question and press send. The company may not be in a position to answer every question it receives during the meeting itself. However, the company review all questions submitted today and publish responses where it's appropriate to do so. These will be available via your Investor Meet Company dashboard, and we'll send you an email to notify you when they're ready for your review. I'd also like to remind you that this presentation is being recorded. Before we begin, we would like to submit the following poll, and if you give that your kind attention, we would be most grateful. I'd now like to hand over to Heike Lundgren, CEO, and David Roth, CFO from 4BASEBIO. Good morning to you both.
Thank you, Mark, and welcome everybody to today's call, and thanks very much for taking time out of your busy schedules today to dial in for our investor update call. So today on the call, it's going to be David and myself from the 4BASEBIO team. We'll be shutting down our cameras once we go into the presentation as well, but then when we get to the Q&A, we'll open it up again. But before we proceed, as you know, we have to take you to our usual disclaimer, so I will just give you a second or two to read through that. Right then, so today we're going to start off with a snapshot overview of the company, where I'll touch on our products and technologies and provide you some further context to the applications where we really think our technologies can have the potential to make a real difference. I'll also give you some insights into the progress we've made in the different areas of our business, and I want to provide you with some guidance on what we expect or what you can expect from us in the near and midterm. David will then take over from me for a few slides and give you a bit of insight on the financial position of the company, and then I'll finally pick up again and actually take you through what I think are the most exciting slides of today, where I'll take you through some technical results obtained with our products and some of the key target applications that we think we can make a difference. So, first things first then, a bit of a snapshot overview. As you know, we are building a life sciences company with two core objectives. So the core premise of the company is to make large quantities of clinical grade DNA that can be used as either a therapeutic agent or as a critical component in the manufacture of cell and gene therapies or mRNA vaccines. As you know, we've developed this proprietary and patented processes and the products that come out of that will enable us to do exactly that. But there is, of course, still quite a bit of work to be done to get our processes to the required scale and obtain our GMP licensing before we really can start commercializing our products. So that's the DNA side of the business. In addition to making DNA, we want to develop a next generation gene delivery platform to safely and efficiently deliver our DNA products into target cells and tissues. That's really to compete with the current viral-based methods, which continue to have significant constraints in terms of payload capacity, immunogenicity, but also in terms of long-term efficacy. So our view is that non-viral based methods, such as the Hermes platform that we are developing, really are the future of safe and effective gene delivery. And the successes that we've seen with the Moderna and the Pfizer vaccines of the viral-based alternatives, such as the AZ and the Johnson & Johnson kind of products, really strengthens our belief that this non-viral approach really will become the dominant kind of force in gene therapy and gene delivery. It's clear that our products and technologies serve a fast growth market with compound growth rates predicted in the mid-30s. So, I think we're really in a great position to benefit from these market trends that are coming towards us. But as you know, timing is essential, and we really have to push on with the build out of our clean rooms so that we can start producing materials on the quality assurance and quality standards that are required by our customers. Therefore, you can expect our headcount to keep growing, and I kind of expect our FTE rate to be around the mid-30s, around 35 by the end of the year. Moving on then to the next slide, I'd like to take a moment to touch on our technologies. For some of you who have been doing some reading on our website, this probably will be very familiar territory. But essentially, core to everything we do in the organization is our True Prime technology. That's kind of the central technology that drives everything we do underneath it. This is the technology that enables us to accurately and quickly create DNA. And it's that DNA that is then further processed into a product which is more suitable for use in the different application areas that we're targeting. You will have seen our recent press release about OSDNA. So we now make two products, the HBDNA, which is a linear closed type of DNA, and then the OSDNA, the new type of DNA that we make, which is an open-ended, an open form of double-stranded DNA, but also enzymatically resistant. So both of those products are now excellent products, but depending on the application that you focus on, one is likely to be more suited than the other. But again, I'll come back to that later in the presentation. So once we've made the DNA, obviously we then need to get this DNA into our target gel, so into our target cells, so that these genes can be expressed. And this is where our Hermes non-viral delivery platform comes into play. As with everything in the world, given that plasmid DNA really was the only available source of DNA, all these delivery systems that have been generated have been optimized for encapsulating this circular supercoiled type of DNA, which is plasmid DNA. The type of DNA we make, however, the linear DNA is very different and behaves entirely different to plasmid DNA. So our objective is to design a delivery strategy that is optimized for the linear DNA products that we create. Now, having said that, the Hermes platform is, of course, also suitable to deliver all types of DNA, and plasmid DNA is part of that, but it can also deliver RNA or even protein. But I would like to stress that for us, our first priority is really to focus on facilitating a non-viral based method for the effective and safe delivery of the linear DNA constructs that we make. So a bit more on OSDNA, perhaps. I'm really excited about this product and the opportunities that it presents, as this product really is ideally suited for mRNA production. And given the current surge in demand for mRNA vaccines, I think it's obvious to everyone that this is clearly a very rapid growth segment for the company. And why this is so important to us is that if you look at how people make mRNA, to make these products, you really need open linear constructs to get clean target DNA transcripts, out of the production process. And this is really why the OSDNA is the ideal product for this application. Combine that with our ability to create highly accurate linear DNA templates of pretty much any size, which on top of that can also incorporate these really tricky bits of sequences, such as like the homopolymeric sequences or the tandem repeats or the ITR that people often want in their sequences. And you can see that we're in a really strong position to become extremely competitive in this market segment. So yeah, we're super excited about the opportunities this presents. Needless to say that, of course, intellectual property is essential in all of this, and particularly in creating shareholder value. So I'm extremely pleased that in the development of this product and this technology, we've been generating really good new intellectual property, which really has enhanced the already strong position we had over our DNA synthesis platforms. And I'm truly convinced that as time moves forward and we continue to invest in our platforms, we will also continue to add strength to our patent portfolio, which is of course very important for future shareholder value. So for our DNA products then, so this slide really is more of an overview slide on where we see the utility and the USPs of the DNA products that we make. And these products, we see really being used either directly as a therapeutic agent. So, that could be like as a DNA vaccine, where, the DNA really is, the therapeutic agent, but of course also embedded in a non-viral based gene therapy. So the Hermes particles that we develop will contain these DNA templates. However, when you look at the mRNA and the AAV kind of opportunities, our DNA there isn't actually going to go into the patient. It's more like a critical ingredient in the manufacturing process of these products, but it doesn't form part of the final composition. And that's really because the DNA is either transcribed into mRNA in the manufacturing process, or it's replicated into a viral genome as part of the AAV production process. That is actually a very important point to make, because it has a significant impact on the quality standard that is required for these different applications. So you can see that where we use our products as a therapeutic agents, the QC and QA requirements are substantially higher compared to using it as a critical ingredient in some of these manufacturing processes. But again, I'll touch a little bit more about this in our technical insight slides further down in this presentation. So our DNA technology is clearly uniquely positioned and addresses really many of the concerns that are associated with plasmid DNA. And that's really because the process is entirely enzymatic, which leads to a much cleaner product, as we don't really have to worry about endotoxins and any kind of bacterial protein or DNA contamination in our DNA preps. More importantly, we can also produce our products much more rapidly compared to what you can do with plasmid fermentation, as we avoid time-consuming steps such as cell banking and fermentation processes. And also our downstream purification is so much more simple, as we have far less volume to produce and far fewer contaminants to remove. So we end up with a cleaner and more efficient process, which has cost advantages, of course, but also has lead time advantages, which our customers highly appreciate. So moreover, the end product on a mass-for-mass basis as well contains more copies of the target gene. And that's simply because if you look at our DNA constructs, we don't have any bacterial elements within that DNA construct. So on a mass-for-mass basis, we can put more copies of the target gene into 1 gram of DNA compared to plasmid DNA. So moving on to the next slide. And this is really a similar kind of slide, but now focusing more on our nanoparticles, on our Hermes platform, which really is a technology that enables us to efficiently package payloads and deliver these payloads very specifically to target cells. As mentioned earlier, we are focusing our efforts on establishing technology that will enable us to package linear forms of DNA, which again, I have to stress, behave completely differently to circular plasmid DNA molecules. But my view is that actually the targeting capabilities of particles will be an essential part in mitigating any kind of unwanted side effects of a potential therapy further down the line. And the exciting thing for us is that the early work we've done so far has shown some really good promise and promising targeting efficiency. So we really have confidence that this technology will work and deliver on its promise. Ultimately, though, for our objective is to enable the delivery of much larger genes than currently possible with AAV vectors. You know, one of the other drawbacks of AAVs is the kind of 4.5 kb size limitation, and to enable a way for repeat dosing strategies so that we can develop therapies that will have a much longer therapeutic effect compared to what currently can be achieved with AAV vectors. So timelines are always important. So, you know, in the next few slides, we'll give you a little bit of an indication on what you can expect from us. Now, 2021 so far has been an amazing year, really, and we've truly made huge strides in advancing the company on so many levels. The talent we've been able to recruit into our Cambridge-based labs has had a tremendous impact on the advancement of our DNA validation and our vector development programs. But also our Spanish team has worked relentlessly to drive scale and efficiency in our synthesis platform. Now, all of that hard work has put us into a great position where we believe we can now confidently transfer these processes that we've developed to a quality control environment. And so we started really picking up the pace in bringing our clean room development and our GMP work in-house, of course, with the help and the support of the necessary experts and expertise and consultants that we rely on to make sure we make the right decisions in these processes. So near term, then, you can expect, well, I think, you know, we need to continue investing, obviously, in the build up and building up the confidence in our DNA products across a wide range of applications. This has to be one of our primary objectives. And as mentioned earlier, we really see mRNA and the AAV production as the areas where our DNA products can make a very early impact. So, you know, our focus has been to work hard on establishing proof of concept data in these areas, and I'll touch more on that in my later slides. But we also see that we can make good impact, and we see a lot of opportunity in cell therapy areas such as CAR-T and of course the non-viral gene therapy, where we think our Hermes platform can make a significant impact. Now, if you look at the field today, it's clear that AAV is currently the delivery mechanism of choice, but the success of the COVID vaccines has given lipid nanoparticles and non-viral based methods some serious pull. You know, these things really do work, and you know, people are now convinced that they have a meaningful place in the clinic. There's also a growing trend towards gene editing strategies, which again is for us a great area to be in, because for gene editing, you need linear templates to insert into the genome, and of course that's exactly what we make. So we think our linear templates will be ideally suited in that application area and that growing market trend. So you can see, the field is moving in our direction, and we're really excited about the opportunities that presents itself for us. On the particle side, we're more focused on creating an optimized particle to package the DNA we make, and we've set our sight on a few tissues and indications where we really believe that we can make a significant impact. And you will have seen our press release with Royal Holloway, with the Royal Holloway project, where we are working on a muscle-based indication, muscular dystrophy, which obviously for us is a key focal point in this area. All of this work, is expected, as with the DNA synthesis platform, is expected to generate valuable IP. So both on the DNA side and on the nanoparticle side, we expect to continue to strengthen our IP portfolio as the development programs progress and continue to advance. Alongside this, we also need to advance the GMP build-out, and I expect that we'll probably complete our first clean room suites by the end of the year, which is really exciting for us. And of course, therefore, I think it's realistic that we can, probably be operational in early 2020, with our first clean rooms. I have to point out that the GMP licensing will, of course, take a little longer, and to some extent, the timing of this is not really in our hands. It's somewhat out of our control, but we do expect to be able to commercialize the product from our clean rooms in 2022 in any event, because many of our customers, will work with us as we go through this kind of licensing approach. So moving on to the next slide. Sorry, I've discussed the near term already, so this is really more like a recap, so you can read this through. But what happens really beyond that is what's interesting on this slide. So it's obvious that I think our first commercial objectives have to be to initiate the sale of DNA products, really in the IVT and the AAV kind of market segments. But in the long run, we expect these revenue streams to lead to a CDMO-style business that will supply DNA across a multitude of applications. On the other hand, we do expect that the Hermes platform has a slightly different path, and as we validate it across several indications, which we intend to take through IND into early stage clinical trials, we would expect that at that point our delivery platform will become highly suited for out licensing opportunities. So, on the one hand, we have a product, the CDMO business, which will sell product, and on the other hand, we have more of a platform technology which we would expect to be licensing as and when the validation has progressed. So, I think, if you look between the lines, I think you can see that we think that within the 4BASEBIO UK SOCIETAS group, we can see two business units with very different life cycles and really also very different valuation metrics emerging. So on the one hand, there's a CDMO style business looking to supply GMP grade DNA products into all these markets. That's highly likely to be high growth, high revenue generating and very profitable business unit. And that, of course, stands in sharp contrast to our discovery business unit, where revenue generation will be more licensing driven, and then the revenue generation is also much further out in time. So, you can expect that business unit to actually be much more cash hungry for the foreseeable future. Anyway, so that's it for my first part. I will now hand over to David, who will say a few words about our financial position, and then I'll take you through some technical slides after that. Thank you.
Thank you, Heike. Good morning, everyone. I have a brief cameo to talk about the financial position and funding, after which Heike will take you through some of the scientific information in more detail. So I've only got a couple of slides, but hopefully they're interesting. I'm struggling with technology already. Oh, apologies, everyone. So financial summary. As you may know, we published our financial results for 2020 at the beginning of June. And these results really reflect the group position following the spin out from 4BASEBIO AG at the end of December 2020. And it's fair to say that while we've reported on this and we're perfectly satisfied with the results, they're not really reflective of the position going forward. But it's worth just touching on them and explaining briefly the context of those results. So the accounting was pretty complex. There's some debate under IFRS 3 how you account for the sort of spin out and acquisition of ongoing activities as we had, and therefore the way we've accounted for this is that we have essentially the full year results of 4BASEBIO SLU, which is our Spanish subsidiary, as well as a very limited stub period for the UK company, which was deemed to be acquired on the 8th of December. And alongside that, there was also just a little bit of activity in the UK SOCIETAS. So really, when you compare that historic period to what we're going to have going forward, clearly there will be a full year results moving forward for both UK and Spain, whereas in 2020 there was only that limited period for the UK. And it also really doesn't reflect the scaling of the lab teams that occurred towards the tail end of 2020, but also more significantly during the early course of 2021. And I guess the most meaningful number coming out of these results is the cash balances that we started 2021, which is approximately 15 million of cash. But necessarily, I think we've indicated before that we are essentially in a pre-revenue situation. We have some revenues which have been brought forward from Spain, and those revenues will continue. And we expect them we don't expect those revenues to grow significantly. We're also not really expecting them to decline very much. So there'll be this recurring revenue, but it's pretty much a legacy revenue and not core to the business going forward. Having said that, and I guess where we are now is our efforts and our ambition is to commercialize our products over the course of 21 and particularly 22, and clearly that may have a significant impact on the overall results and performance moving forward. But assuming that we remain relatively static in the near term on our legacy revenue position, the group inevitably will be in a significant cash burn position. And as a result of that, we would expect our cash runway to last well into 2023. And then beyond that, I think as we've reported as well, we have access to a £22 million loan facility. So clearly we are in very good financial shape and well placed to make investments necessary to commercialize technologies, to invest in our team, and also make whatever necessary CapEx expenditure that we require. So having said that, I just want to turn briefly to funding to just explain where we are in terms of our thinking. So because of the combination of cash and loan facility, no funding and no fundraising is foreseen, and certainly nothing is planned. And so the question really is, in what circumstances might we look to raise additional capital? And I guess perhaps before answering that question, the most important point is that any capital raise would need to be at a share price or a market value with which the board is comfortable. It's hard to say what that would be, and I guess it really depends on the situation at the time. But I think it's fair to say that we'd be looking for very meaningful appreciation on the current share price before we would contemplate any sort of fundraising. And then in the circumstances where we might then consider it is if there's a very significant capital investment needed, and ideally that would coincide with a value inflection point. So, for example, a large-scale GMP facility or ongoing investment into indications that are moving into clinical trials. And then another scenario might be where we think it makes sense from a strategic position and from a valuation position to bring in a specialist institutional investor to offer validation of the company valuation and the technology. But I'd reiterate that we've no plans for a capital increase at this time or for the foreseeable future, and we expect to continue to proceed entirely within our available resources. So that's really my cameo. I'll pass back to Heike now, who will take you through some of the inside slides.
Thanks, David. Right, so those who've been listening to my slides and my pictures, have heard me talk a lot about the strengths and the benefits of our technologies. and the products that we make. So in the next few slides, I'd really like to put some meat to the bone there with some data validating some of these statements that we've been making, and also showcase how our products are performing in these key application areas that we've identified. What I really want is that the main take-home messages from this is that, you can see that, A, we can really make high-quality linear DNA products, and B, that these work extremely well in IVT-based mRNA production and AAV production. So here we go then. This first slide is really more of an introductory slide, and it kind of shows you that we can make DNA constructs, sorry, and that the amplification process that we have typically yields us DNA at a concentration of 1 gram per liter. So if you look at the slide and at the graph, you can see that, somewhere around 0.9 and just over 1 milligram per milliliter or gram per liter is kind of the DNA output that we get. Now that's actually about 1,000-fold higher than what you typically can achieve on fermentation-based approaches. So that's meaningful in terms of bringing our size and scale down and bringing our costs down. I've also put in some tangible data on the fidelity of our application, which shows that, our process really is about 200,000 times more accurate compared to the conventional PCR-based approaches. So again, the overall conclusion from this slide is that we can make top-quality DNA at a really high concentration. Moving on to the next slide, this really shows the ability or our ability to make small and longer DNA constructs. So what you see here is an agarose gel. So the gel really just functions as a molecular sieve, and the smaller DNA pieces will migrate deeper down gel compared to the larger pieces, which are retained higher up in the gel. So a construct of a given size will therefore condense into a discrete band on the gel. And on our gel here, you can see that the smallest construct is around 1,800 base pairs, so 1.8 kb is kilobases, so 1,800 base pairs. And the largest one is around 11,900 base pairs. Now, this is just a snapshot of what we do. We can make smaller, we can make bigger, but you can see, we're very able to make a wide range of sizes of products. Also, I think what's also very important on this slide is that, we can make clean product. So you can see that on the gel that there really is just a single discrete band, so there's no contaminant or fragmented DNA present above or below the target bands. So from a DNA perspective, this shows you that we have a very clean DNA product. And of course, with the fidelity that you've seen on the previous slide, you also know that the DNA construct will be highly accurate and will be the right sequence that the customer is wanting. But as you know, our processes are enzymatically driven. So for us, it's critical that we can also demonstrate we can remove these enzymes from the final DNA product. And to prove that we have removed these protein contaminants from the DNA preps, we run a different type of gel. So it's essentially still a molecular sieve, but now it's designed to separate and visualize the proteins, the enzymes, rather than the DNA. And I'll take you through that on the next slide. So on the left-hand side here, what you can see is a Hermes stain gel. So Hermes is a blue dye that likes to bind proteins and makes them blue. So what we've done here is we've loaded 2, 4, and 6 micrograms of purified DNA. And what you see is we cannot observe any protein bands on the gel. So if you look at the first lane of the first column, or U, you can see there's a few bands there. So these are essentially protein bands. If you look at the 2, 4, and 6 microgram lanes, there's essentially nothing there. Now, this allows us to estimate that our purity is in excess of 98%, because we would expect to see a 100 nanogram band. So if you divide 6,000 nanograms or 6 micrograms over 100, you get that kind of purity. If we use the same approach now, but we use a stain which is actually much more sensitive, like silver stain, again, you can see that we can't see any kind of bands on the gel that correspond with the enzymes we've used in our production processes. So in the pre-purification lane, you can see a lot of protein bands. In the purified lane, you cannot see any protein bands. The only protein you can see is the DNase one that we've actually had to add to these samples to digest the DNA, because DNA also stains with silver and would generate a lot of background that would prevent us from doing the analysis. Now, again, of this slide, we can estimate a purity of around 99%, or actually in excess of 99%. So you can see that overall, the take home from this slide is that we can very efficiently remove our processing enzymes from the final product and deliver a very clean and pure product to our customers. So I think the first two slides show you that we can make good quality, high quality, very pure DNA. Moving on now to the application area. So the first one here is just a bit of an overview of our DMD collaboration. So I think it's very important to provide some context on the DMD collaboration with Royal Holloway, and why we think this indication is really ideally suited for our technologies. For those of you who are not familiar with DMD, it is an X-linked inherited monogenic disease caused by mutations in the DMD gene, and has an incidence rate of about 1 in 5,000 male births, as it is on the XY kind of genes. The DMD gene is actually one of the big ones, with a DNA sequence coding for dystrophin, so the protein that has been compromised in the affected people, being about 12,000 bases long. We know AAV vectors can only pack 4,500 base pairs, so you can see that there's a significant problem here. So to make an AAV strategy work, people have to cut back on the payload, and that results in exon skipping strategies, or delivery of microdystrophins, which is essentially an abbreviated version of the real protein that is required. Now, the problem with AAV vectors as well is that you really require very high viral titer. So that's the concentration of viral particles that is used to dose to people. And that's really required to achieve the desirable level of transduction and getting enough transgene into the muscle cells. This is caused by the fact that the AAV vectors can really only be dosed once because of their immunogenicity issues. And of course, these high viral loads that are required often lead to toxicity and adverse effects, then again compromising the success of a potential treatment. So our objective is now to create a Hermes-based DNA vector that can deliver a highly effective patented full-length dystrophin gene, specifically to these muscle cells. And due to the preferable immunological properties of our Hermes vectors, at least compared to viral vectors, we anticipate that we could really enable repeat dosing strategies, and that, of course, in turn will then benefit from low toxicity and adverse effects and create the possibility for prolonged therapeutic effects. Now, developing and validating these vectors is unfortunately not quick. It's a long process, but I think it'd be useful for us to provide some indication on what we think the next steps will be in this project. So currently, we are validating the vector with a reported gene in vitro cell line. So this is just in our labs here in Cambridge. But the real objective is to demonstrate that we can specifically and effectively deliver the transgene to a muscle cell. Once that's completed, we anticipate to move towards in vivo animal validation of this vector, and I expect that that probably will be completed around the year end. I think that will then put us in a great position to evaluate a therapeutic vector, so one actually incorporating the DMD gene, and initially we'll do this in a single dose study, and that will then be followed up with a repeat dose study in 2022. So you can see there's a lot of work still to be done, but the early work is moving through very nicely. And as and when we move through these stages, we'll of course keep you informed and update you about the progress that we make. So now into the application areas where we think the DNA can make some early commercial impact. And I think this slide is really quite nice as it demonstrates the utility of our OSDNA products in mRNA production. I think by now you all know the value of mRNA and what these products have achieved in terms of beating the COVID pandemic, as both the Pfizer and the Moderna vaccine are essentially mRNA products. And this success in vaccines will really drive the demand for mRNA, with some vaccine makers expected to require potentially maybe even 100 kilograms of DNA per annum. So, I mean, these quantities are actually really hard to get your head around, and definitely at this stage, the global production capacity of DNA is nowhere near that level. So there's clearly a huge amount of growth in that segment. We estimate that the cost of DNA for a single vaccine dose will probably be in the region of 50 cents to maybe a few dollars. But evidently, where 1 billion doses are required, the market opportunity is clearly very, very significant. So the exciting thing about this slide is that, in our hands at least, our linear DNA products are clearly outperforming the current source of DNA, as in the linearized plasmid, and we can achieve about 10 to 8 times more target DNA compared to plasmid DNA. Moreover, we also observe that our mRNA preps are a lot cleaner compared to what we can achieve with plasmid DNA, which we think is again due to the higher purity of the DNA products that we make. So the single band of the that you can see on the agarose gel and the lack of protein contaminants, which I explained a couple of slides ago, is really working in our favor here. And obviously, we now need to get this product validated in the hands of potential customers, which is the next stage of the development we're now entering in this project. Moving on to the next slide. So these next two slides are really more about AAV. What I really want to showcase here is the excellent performance that we achieve with our DNA products when we use them to make AAV particles. That other lower hanging fruit commercial opportunity that I've talked about today. So AAV, as said, is really the primary choice of vector for gene therapies at the moment, and there's many clinical trials on the go at this point. And the cost of a single dose of DNA for an AAV therapy can vary quite significant, but you can see, somewhere between $75,000 and $10,000 per patient is quite significant. Now, if you look at the chart, the blue bars represent transfection efficiency, and that is essentially a measure of how easily we can get our DNA into the AAV producer cells, so the cells that make the particles. Now, the data shows we can obtain similar or maybe even slightly better levels compared to plasmid DNA, but what's really exciting is the red dots, which show that our constructs have higher expression compared to plasmid DNA, so more protein is being expressed. Here, for both experiments, we use the same mass dose of DNA, and as I explained before, compared to plasmid DNA, we have more copies of a transgene in 1 gram of DNA, and we attribute this effect, really to the increased expression that we have on that basis. On the next slide, you can see, and this is actually commercially more relevant, I think, than the previous one, because this really, the first slide showed really that our DNA was suitable for the production of AAV, but on this slide, I think what we show is that the AAV particles that we make are actually better and of higher quality than the ones that you could obtain with plasmid DNA. So when AAV particles are assembled, many of the particles actually don't contain a viral genome, so there's a lot of empties around, and of course there's some full ones around too. But the packaging of DNA into particles is really inefficient and leads to very low full empty ratios. So what we've done is we compared the full empty ratios of the particles we've made with our DNA products to the particles that we've made with plasmid DNA products, and we noticed a substantial difference in the full empty ratio, with plasmid DNA performing only half or about 30% as good as what we could achieve with the HB DNA products. So that's really quite exciting. Now, when we then did some further work, this was further corroborated when we looked at the infectious dose, and that is essentially the minimum dose that you require to infect a population of cells. So it's a measure of potency of the particles that you've made. Now, obviously, the lower the dose, the better, as that means it's a more potent particle, and that you can deliver your transgene more effectively with that. In our experiments, the TCID50, which is the measure of the infectious dose, was about 1 or 2 logs lower, so about 10 or 100-fold lower compared to what we achieved with plasmid DNA. So this shows that our DNA products generate particles of higher quality and higher potency, and that really strengthens our belief that our products have a really good role to play in viral vector manufacturing, and we're working, of course, hard now to progress our AAV program to the next level of commercial validation. So, in summary, I hope you got the vibe that I feel the company is in excellent shape, and I think we're really well funded to achieve our objectives. And as David mentioned earlier, any future capital increase is entirely optional, but moreover, at the current share price, we don't really think it's appropriate. But we've worked hard to recruit additional scientific talent into the organization, and we'll continue doing so, of course, as we expand our operations. But I really feel that now we've got a very strong core scientific team, and I'm confident that our scientists will deliver and pave the way for the commercial phase of our products and technologies. Consequently, I think you can see some strong value inflection points. Well, I can see some strong value inflection points around Horizon, which I hope will be appreciated by the market, of course, in due course. But personally, I think the work that we'll be doing in the next 6 to 8 months to firmly establish our clean room-based DNA manufacturing capabilities, along with the strengthening and the expansion of our proof of concept across a wide range of commercially relevant DNA applications, will have significant impact on the future financial performance and ultimately the commercial success. And as always, we will keep you abreast of our developments as and when they materialize. So I think that's it for me. I'd like to thank you all for listening, and we'll move on to any kind of questions as they may have come through.
That's brilliant. Heike, David, thank you very much indeed for updating investors this morning. Ladies and gentlemen, please do continue to submit your questions using the Q&A tab situated on the top right-hand corner of your screen. But just while the company take a few moments to review investor questions submitted already, I'd like to remind you that a recording of this presentation, along with a copy of the slides and the published Q&A, can be accessed via your Investor Meet Company dashboard, and we will send you an email to notify you when they're ready for your review. I'd also like to remind you that your feedback is important to the company, and immediately after the presentation has ended, you'll be redirected for the opportunity to provide feedback in order the company really understands your views and expectations. Hi, David. Obviously, the Q&A tab on the right-hand corner of your screen, if you'd be so kind as to open that. If I could ask you to, where appropriate, read out the question and then give a response where it's appropriate to do so, and I will then pick up from you at the end. I don't know if your cameras are on, or there we are, perfect. And if you need the slides, obviously they're there for your use as well.
Sure. Thanks, Mark. So the first question I'll just read out is, can you expand a bit about the size of the company, please? Number of employees, office locations, and what have you? So, gosh, where do you start with this? So, in 2020, we were obviously a spin out from 4BASEBIO AG, which is now 2Invest AG, and really at that point in time, the UK operation was essentially a service function of 4 individuals, and we also had a significant research presence in Spain. And then during the course of 2020, we started to expand our base in the UK back to where we were previously. And I suppose as part of the former life, we actually owned a building here near Cambridge. And in September 2020, we basically took possession of that again. So it's a freehold space of 12,000 square feet where we have labs, warehousing, and what have you. And we've essentially split our operations into 2. So in Spain, in our research facility in Madrid, we focus primarily on platform research around DNA. And also, there's a real expertise in terms of enzyme manufacture and enzyme development. Here in the UK, we are focusing more on nanoparticle development and also the validation work that comes alongside that in terms of our DNA. So at the moment, we have around 27 employees, which is split about 10, 11 in Spain and the remainder here in Cambridge. And then moving forward over the course of the year, there's going to be a little bit of additional recruitment in Spain, but the principal focus is here in terms of filling out our scientific teams, particularly around the validation work and that Heike mentioned. So we expect to move, and clearly depends on recruitment timetables and whatever else, but we expect to move from around the 27 we have at the moment to beginning of next year around 30 to 35 staff overall, of which around 2/3 will be based here in the UK. Second question, what protection do you hold over the technology you have? I think as Heike's mentioned, a very significant value consideration for us is the intellectual property and the protection we have around those products, and clearly that's pretty fundamental in terms of life sciences and the capacity to exploit that intellectual property. So we have patents across the range of products that we have. And we continue to create new IP as we validate and develop our products, and as we continue our research both in Spain and here in the UK. And that's really a key consideration for us, because clearly alongside the capacity to manufacture DNA and also develop our nanoparticle technology, it's also the opportunity of what that technology could be worth to others. So what's it worth in terms of licensing around our nanoparticle technology and the IP value that sits within our DNA. So that's a key consideration for us, and clearly we spend a lot of time focusing on that intellectual property.
Yeah, I mean, if I can just touch on that one as well. I mean, we're spending a lot of resources with patent lawyers at the moment. So, we've announced one IP filing recently as well. But with all the work that we do, and it's probably not going to come as a surprise that we're generating a lot of new inventions, and we're reducing these to practice, and we expect those to generate new patent filings in due course as well. So yeah, I think as David mentioned, there's a lot of existing intellectual property, but there's a lot being added to it right now as well.
Moving on to the next question then, what do you view as the greatest risk to your short-term goals? I mean,
for me, it's probably talent recruitment.
Yeah.
I mean, we've got really good people that we've been able to recruit, but of course we still have massive expansion plans, which means that we need to expand our teams along the way as well. So continuing with recruiting the best people is challenging, and the rate of development and the pace of development that we have, makes this probably one of our biggest challenges personally.
The next question is, how does HPDNA and OSDNA compare to doggy bone DNA from Touchlight? They're both synthetic products. I think as Heike alluded to in the presentation, the demand for DNA going forward is expected to increase exponentially. And our view on this is that there's certainly more than enough space in this market for one or more manufacturers of synthetic DNA. And we're very pleased that they're in here, because to some extent they are pathfinders for the synthetic DNA market.
Yeah.
We've got a couple of questions, but I have to take the second one first. What would be required for HPDNA or OSDNA to replace plasmid DNA? Is it possible to replace plasmid DNA for a marketed product, or would that require reapproval of that product?
Well, I mean, let me just touch on this for a minute, and it really very much depends on the application that you're looking at, right? So, if you're using the DNA as a therapeutic product, so if it's a DNA vaccine or it's embedded within a non-viral vector and the product itself goes directly into the patient, I think that would be quite challenging to replace any DNA source or any ingredient from that preparation for something else. So really you need early stage work there and be embedded from the discovery phase all the way into a commercial product. Now, when you're looking at AAV manufacturing and mRNA manufacturing, that's entirely different. Your critical ingredient, and those critical ingredients, and we believe at least, are more readily to be swapped out as they ultimately don't impact on the final product, because they're not incorporated within the final product. So, in terms of opportunity there in AAV and mRNA production in particular, we really see a lot of opportunity to replace plasmid DNA and in favor of HPDNA and OSDNA products.
And the second question is the company on track to meet its goal of 1 kilogram of GMP manufacturing capability by year end? Would that cover both HPDNA and OSDNA, or just HPDNA? So I think, as we've described, we're in the process of developing and building out those clean rooms, and with a fair wind, we expect to be there by the end of the year. And those clean rooms would certainly give us theoretical capacity to do this. Whether in practice it's something that we can or would do is a second point, because clearly that's about the market demand and where we are in terms of those customer relationships and what have you. But certainly would have the capacity to manufacture both HPDNA and OSDNA.
David, Heike, I might just jump in, obviously on the basis that I think you've addressed every question that's come in from investors. So thank you so much to the investors for submitting those questions, and indeed to you both for taking the time to give your responses. Thank you once again for updating investors also. Could I please ask investors not to close this session, as you'll now be automatically redirected for the opportunity to provide feedback in order that the company can better understand your views and expectations. This will only take a few moments to complete, but I'm sure will be greatly valued by the management team. On behalf of the management team of 4BASEBIO, I'd like to thank you very much indeed for attending today's presentation. That now concludes today's session. Good morning to you all.